welcome everyone to Introduction to Epigenomics. I'm very excited to be here with you today. Um, just a little bit of housekeeping before we get started. Let me just get these Zoom controls out of the way. We're going to talk about a lot in this class. So I have a very full agenda full of really exciting things. But I don't want you to get too overwhelmed because we've got places that we can cut if we need to. So we've got sort of on day one an introduction to epigenetics, sort of a primer and motivation for studying that, how to measure it, and some quality control and sort of getting into the epigenetic association studies. And then we'll also talk about the computing environment in RStudio, sort of get you set up to do a little bit of analysis around epigenetics. We do have the course set up right now where you'll sort of be doing the coding kind of as we're going through the slides and explaining the results, but we can also take, a little break here and there to let you run a little bit of code if it starts to get, if the code is running a little slow and you need a little time to catch up, you can just let us know and we can pause for a minute. People can catch up on the code. In the second day, we'll have some epigenetic association studies, especially epigenome-wide. We'll talk about a lot of pieces there. We'll talk about running and plotting those association studies, talk about multiple testing. We'll talk about what you do with those results after you run the association studies. So in terms of kind of looking at what are the biological implications of things that you find or what have other people found with the epigenetic sites that you found that are interesting. And then we'll look at different ways of doing sort of an epigenetic analysis. For example, looking at big regions of the genome at once. And then on the third day, we'll focus more on kind of summary statistics. So looking at what we call epigenetic clocks, polyepigenetic scores, and surrogate biomarkers, which are kind of summaries of the methylation patterns across the genome. If we have time, we'll talk about genetic and environmental influences on methylation and some additional resources. But I did try to weave those topics kind of into other pieces throughout the course. So if we run out of time for that part of the lecture, no big deal. We will have a homework assignment every evening, including on the last day of class, but we'll provide solutions to those. So once we give you the homework assignment, we'll go over the solutions to those the next day in class. And then on the last day of class, we gave you slides already that have all the solutions there. In this class, epigenetics is a really big topic. There's a lot of different places you can go with epigenetics. I'm going to give sort of a primer and motivation for studying epigenetics, but in the class, we are mostly going to focus on DNA methylation, which is one type of epigenetics. We're mostly going to focus on that in human populations. So you can study the epigenome of many different species, but we're going to mostly apply that to humans. And when we do some of the quality control and things like that, we're going to be doing it on microarray data. There are other ways to measure epigenetics, not just microarray, but a lot of the concepts and topics that we learn in the class can be applied to other types of epigenetic data as well, no matter how it was measured or even what species you're applying it to. So hopefully if you're not working with that exact setup in your own work, you'll be able to apply a lot of the concepts that you learn in the class to your work. So I just wanted to, before we start, just kind of give you a sense of where we're going in that direction in the class too. Okay, so let's start just by talking about a primer on epigenetics. Really, what is it? So remember that in the genome, we have DNA, the DNA sequence, A, C's, T's, and G's. It is 3.2 billion bases in the haploid cell. So you get 3.2 billion bases from your mom and 3.2 billion bases from your dad. It's 46 chromosomes, about 20,000 genes, and it's about 2 meters long. And a typical cell is about 10 to 100 micrometers. And so you have a very, very, very long, very thin piece of DNA, and you're trying to pack it into this tiny little cell, right? And so the packing structure of the DNA is very complex and it's necessary structurally. It creates a lot of challenges, but it also creates a lot of opportunities for the DNA to kind of come together in different ways to be regulated. And when we talk about regulation, we're talking about regulation of the expression of genes, right? So just to kind of like visualize this a little bit more, let's say you have like the DNA double helix, all those A's, T's, C's, and G's, and the DNA is actually wrapped around these proteins called histones, which look like beads on a string. It's like the DNA is kind of wrapped around, and then it goes to the next one, and then the next one. These beads on a string then pack together again, and then fold together again in this loop structure, and then they keep condensing. And that's what kind of makes up your entire chromosomes. And the chromosomes actually interact with each other. And the way that those interactions are orchestrated is actually by the epigenome. So here you have the DNA, the chromosome, sort of laid out in a karyotype, essentially, where you have the chromosome 1s are together, the chromosome 2s are together, and 3s are together, and they're stained here different colors to show you each chromosome so you can identify it by its color. And this is what it looks like when they're all laid out nice next to each other. But what does it look like in a cell? Well, this is kind of what it looks like in the nucleus of a cell, right? So you can see the different chromosomes are kind of like unpacked a little bit. They're kind of like messily spread out in a way. And they're sort of like touching other chromosomes in really specific ways. And actually, you can look at this map and you can kind of map out which chromosomes are touching which other ones in the cell. And this is a very characteristic pattern. So this is not by accident. And like I said, this is orchestrated by the epigenome. And what it does is it allows not only pieces of the same chromosome to fold on each other and interact with each other, but also pieces of other chromosomes to interact in really specific ways with, a different chromosome that's entirely not anywhere near it in the genome as a linear genome. Okay. All right. So why is all of this important, right? The study of epigenetics. What is really epigenetics? So the epigenetics, really when we talk about that, we're talking about the regulation of the activity of genes or the expression of genes that's not dependent on the sequence of DNA, right? So we know, like if you get mutations in your DNA, that can cause disease, but also if we have changes in the way that our genes are regulated or expressed, if they're turned up too much or down too much, if they're turned on at the wrong time or in the wrong cell, that can also cause disease, right? So the epigenetics here, they're important for regulating the protein and the RNA levels in cells, right? That's the expression of genes. It's the expression of RNAs that even don't code for genes. That's why it's implicated in human disease. And what's really interesting about the epigenome too is it's influenced by both genetics and the environment. So there's just like genetic mutations are associated with disease, but sometimes the reason why is because it changes the expression of a gene, right? Well, sometimes it changes the expression of a gene through its modification of the epigenome, right? So genetics can influence the epigenome, but the epigenome is also really influenced by environmental factors. So things like toxicants, like air pollution, things like smoking, health behaviors. things like stress, social factors, socioeconomic status. All of this stuff can influence your epigenome too. And so this is just a different picture of the epigenome. This is your chromosomes. This is chromatin, which is kind of like when you have the DNA that's like a little bit spread out in the cell. And then these are the histone beads on a string. And this is the DNA. And then here's a methyl group that's on the DNA. So this is showing DNA methylation, which we're going to talk about in more detail too. Another really important thing is that tissues in your body are differentiated by epigenetic patterns. So you've got the exact same DNA basically in every cell of your body, but all your cells look different from one another. You have skin cells and eye cells and liver cells, all kinds of cells, right? How did they get that way? Well, they get that way because their epigenomes are different, right? Same DNA, but they're expressing different genes at different times, right? And so what happens is you start with a stem cell, which can turn into any different kind of cell in the body. And that stem cell goes through a series of decision points and ends up being ultimately some kind of cell in the end, a specialized cell. And again, that happens through the changes in the chromatin, the way those interactions happen. And that's through the epigenome. And so this epigenetic specificity leads to specificity in the expression of the genes, right? So we'll talk more about this in a minute too. And what are the mechanisms that influence the epigenome? Well, there's kind of three types. So one is DNA methylation at CpG sites. And this is the one that we're going to talk about and study the most in this class because it's easiest to measure. So we know the most about it. And most people are studying it who study epigenetics. That being said, there's a lot to learn from the other type of epigenetic mechanisms too. The next one is called histone modifications, and the third type is called non-coding RNA or regulatory RNA. So all of them are shown here. We're going to talk a little bit more about each of them, but essentially DNA methylation is a methyl group that's added to the actual DNA strand itself. The histone modifications are the beads on a string. They actually get like all of these different adjustments that get made to their the tails of their some of the proteins on the histones. And then the non-coding RNA is actually really interesting. So one example of it is the microRNA, where you have a piece of RNA that's not eventually destined to make protein. It does a different job in the cell. So this particular kind of RNA, it forms this like hairpin loop structure. So it folds back on itself. It's exported out of the cell. It's cut and then it's denatured. So it separates one strand from the other. And then it targets a messenger RNA that's on its way to make a protein. And it targets it, and another complex comes along and degrades that target RNA. So all of these actually modify the expression of genes in different ways, right? So the methylation is going to control expression of genes that are really close to that methylation site usually. The histone modification is going to affect like a little bit bigger region, like because this is wrapped around the histones, right? So it's a little bit bigger. And then this microRNA can actually affect something really far away in the cell because this target messenger RNA might be made even on another chromosome. So if we just take a little step back and think about the influence on gene expression, just to like make sure everybody remembers and is on the same page, remember in the nucleus, you've got the DNA, and you have both exons and introns in the DNA. So if you remember, Exons are the part that codes for a protein, and introns are a piece of DNA in between the exons. And the introns get cut out, and then from that, you make what's called messenger RNA, right? That's the part that, well, with the messenger RNA, the introns are cut out, and then that messenger RNA is translated into protein. So the DNA is transcribed into messenger RNA. The messenger RNA is translated into protein. That's kind of like the central dogma of DNA, RNA to protein. And so that control of protein production via this RNA is critical to the activity of the cell, right? And so all of these different pieces, the histone modifications, the modifications to DNA, and then the non-coding RNA are going to all affect the expression of genes. And this one I'm going to actually skip because I'm going to cover it in a second. So any questions on that before we move on to more specifics about the mechanisms? Okay. So just to be clear, so DNA is made up of these sequences of A's, C's, T's, and G's, the nucleotides or the DNA bases, right? You remember the double helix, A pairs with T, C pairs with G. And I just wanted to remind you of some nomenclature in genetics. So remember you have SNPs, which are single nucleotide polymorphisms. So this is a location in the DNA when you have different options for the DNA base present, or different alleles or different versions. Okay, so maybe right here, this person, or sorry, one chromosome could have a T here, like this one does, a TA pair here, and maybe another chromosome at this exact location would have a C and a G, right? So the first one would have a T allele at this location, and the second one would have, like, say, a G allele at this location. Or C, whatever I said. Two different alleles. That's a single nucleotide polymorphism, or a single nucleotide variant, which is the same thing, but just when one of those versions is very, very rare in the population. So when these words come up later, you won't be surprised by them. It just means a different version of a base at a particular location. When we think about the DNA regulatory regions, one thing that we have to remember is the genes, which have their exons and their introns, right? Coding for protein here. These are going to be cut out upstream of where the protein coding region starts. So remember, DNA is oriented in a 5' to 3' direction. When we say upstream, we mean 5', and that label of 5' to 3' is just based on how the backbone of the DNA is oriented. So there's a sugar phosphate backbone to the DNA, and it's an orientation of the sugar molecule there. So when we say 5' to 3', we're just referring to that. So upstream, before the start of the gene at the 5' end, there's a region called the promoter, and that's where the machinery that transcribes the DNA into RNA is going to come sit down on the DNA, and it's going to start making that it's going to start transcribing that piece of DNA into messenger RNA, right? And remember that the promoter region of the gene is really important because it really is the place where it's going to regulate whether that gene is transcribed or if it's not. So if those transcription factors can't sit down on the DNA, then they can't make the messenger RNA from that gene, right? And we won't get any protein from that gene. So the promoter is really important regulatory region. And then other places in the genome can be what we call enhancers. And the enhancer can actually loop around and touch the promoter. So sometimes the enhancer's on the same strand of DNA, but farther away, and it just forms this loop and comes back around. And it also has transcription factors that work together with ones that are touching the promoter, and that even enhances and says, yeah, you got to transcribe this gene even more, right? So the enhancers and promoters are both really important regulatory regions. And sometimes enhancers can even be on a different chromosome. So that's the central dogma, and gene expression is initiated by these proteins that bind, right, to the enhancers and the promoters. Okay, one of the mechanisms we want to talk about is DNA methylation, right? So DNA methylation happens on cytosines or C bases in humans. Cytosine can be unmethylated or it can be methylated. It's just the addition of a CH3 group to the C base. It doesn't change the base. It's still a C, but it has this extra methyl group on it. Okay. It happens when you have a cytosine that's 5' to a guanine. So you have going along the DNA 5' C, G, the 3'. Those are the places where it can happen at. We call these places CPG sites. The P just stands for phosphorus because DNA has or phosphate because DNA has a sugar phosphate backbone. So you've got C and then phosphate and then G in the genome. So these CPG sites, we're going to talk about those a lot. So in any given cell, we have 2 copies of every base pair sequence, right? You have one chromosome, one from your mom, one chromosome, one from your dad. And at those places, you can either have at some particular CPG site on your mom's DNA, it could be methylated or it could not be methylated. And same for your dad's, right? So in a given cell, you could have 0% methylation at that CPG site. You could have 100% methylation, or you could have the one from your mom is methylated and the one from your dad is not. And that would be like a 50% methylation, right? And CPG sites are often clustered into what we call CPG islands. So you have these islands where you have a lot of cytosine followed by guanine bases. If you look at the entire genome, the whole genome is actually depleted for CPG sites. So much less than we would expect by chance alone. But there are places in the genome where they're really, really enriched in these small areas, and they're called CPG islands. Those are important because the regions that are just outside these islands are called shores and shelves. And on the shores and shelves, there are still CPG sites there. They're right around the boundaries of these CPG islands. They're about like a couple kb upstream and downstream from these islands. Those are really important, we found, because they're really activated a lot or different across different tissue types. So they're really important for tissue specialization. So these CPG islands are almost always like methylated. Most of the time they're methylated. But what's going on in the shore and shelf seems to be really important. So we'll talk more about that later when we identify CPG sites that are associated with disease. If they're in a shore or shelf, we're like, oh, that seems like it might be more important for regulating nearby genes. This is an animated slide. Let me see if I can do this. So unmethylated CPG sites and gene promoters specifically usually allow that gene to be expressed. So this is a promoter region of a gene, and the transcription factor can come sit down on it, right? And then it can, the RNA polymerase will come and it will make the messenger RNA. So that's what usually happens if the promoter region is not methylated. If you have methylation in the promoter region of a gene specifically, it usually leads to repression of gene expression, right? So here's another promoter region, has methylation on it. That actually stops the transcription factor from sitting on it. And so it kind of tries to sit and it can't, and it just goes off into somewhere else, right? And no gene is expressed there, right? So we know in promoter regions, methylated usually means less expression, and unmethylated usually means more expression. Now, in other regions of the genome, methylation is also going to regulate expression, but the effects really aren't as predictable. We know that the first exon is really important. A lot of times there, more methylation means actually more expression, but most of the regions of the genome are difficult to predict. All right. The level of methylation is sensitive to both genetic, sequence, and environmental factors. So exposures can influence DNA methylation and gene expression. This is just an example. In mice, when you have maternal mice fed high-fat diet versus a low-fat diet, and then you look at characteristics in their offspring, you can even see lower DNA methylation and higher gene expression, for example. So we know exposures can influence DNA methylation and subsequent gene expression. There are a lot of normal roles of DNA methylation in the cell. So they help control development. They actually inactivate the X chromosome. So remember, in females, you have 2 copies of the X chromosome, and usually we only need the dosage of genes from one of them. And so one of them is randomly deactivated. That's done by methylation. Gene imprinting, where you have spaces in the genome where you only want a gene expressed either from mom or from dad, maternal or paternal copy of the DNA. Methylation can turn off the one that you don't want expressed. It maintains the chromatin, which we talked about already, and it has that tissue-specific expression control, which we talked about. Okay, so that's DNA methylation. For histones, we talked about this beads on a string. So the histones are made up of multiple proteins, and they have these tails sticking off of the protein. So they're pieces of the protein, but they're like the ends of the protein, and they sort of stick out like tails. There's a lot of different modifications that can happen to these tails. There is, for example, when you have histones that don't have acetylation in the tails, they tend to like bunch together. But when you put acetylation on the tails, it changes the electric charge, and all of a sudden the histones separate from each other. That separation of the histones actually opens up the DNA in that region and allows those transcription factors to come sit down on the DNA and transcribe. So this is what we call like closed chromatin, and this is open chromatin. So genes here will be expressed and genes here will not. And the histones actually have a lot of different modifications that can happen to them. These are the different histone proteins, H2A, H2B, H3, and H4. And these are some of the different modifications that can happen. So this is acetylation, there's ubiquitination, phosphorylation, methylation, all of these different ones, okay? These all are going to alter expression on a set of genes. Again, doesn't change the DNA sequence, just opens and closes that chromatin. Histone modifications also are very correlated, again, with gene transcription. These are just showing you the different histone proteins, H2, H3, and 4. These are different places on the histone tail and different modifications that's done to them. So like this is the methylation, for example. This is the ubiquitination. And it's showing you that like there is this big correlation with the rates of transcription and sort of where that's kind of opening and closing the chromatin. But one thing I really wanted to point out is that DNA methylation and histone modifications, they work together and they reinforce each other to regulate the transcription of genes. And so none of these things are kind of working in a vacuum. They really are working together to regulate in a certain region. Okay, regulatory RNAs, these do not code for proteins. So again, this is a functional RNA molecule that's transcribed, but not translated into protein. So we used to think that RNA was only for making messenger RNA, but now we know there's all kinds of other kinds of RNA in the cells that do all kinds of different jobs. So there's that microRNA I talked about. There's also two other kinds of RNA that silence genes in a sequence specific manner. One that actually just cuts them, and then another one that regulates through a variety of other mechanisms. So they're doing a lot of different things. And so this is another kind of epigenetics. Okay. Another few words about tissue specificity and timing that are going to become important as we think about our analysis. So tissues, again, have different epigenetic profiles. So if we look at samples from two different tissues, this is the correlation pattern between samples of placental tissue and samples of blood. So these are correlations within those tissue types and then across. So you can see that there is some correlation in the DNA methylation patterns between blood and placenta, but it's definitely not as strong as the within tissue type. And that correlation with another tissue type, which is semen, is almost like non-existent. Here's one reason why it becomes interesting. So when we look at things like how do the methylation and correlation patterns differ across these tissue types? Well, this is an example from brain, liver, and spleen. And this is looking at methylation patterns in a gene. And it's mapping here the CPG density. And so it's showing you a CPG island. See how many CPG sites are in the CPG island? It's a very dense region of CPGs. And this is offshore. This is offshore, right? This is the shore of the CPG island, right? Which I told you is an important area for differential tissue expression. Okay. So the brain, liver, and spleen look very different in the shore site. They look really pretty similar in the island site at this one gene. So if you look up the expression of this gene, you can see that the spleen and the liver actually have pretty low expression levels of the gene. The spleen seems to be the lowest. The liver is pretty low, but the brain actually has a high expression level of this gene. And so here, the shore has a very different methylation pattern in the brain. And it's reflected in that expression of this gene. And again, because these cells are differentiated by epigenetic patterns, what we need to be conscious of when we're doing analysis is that when we take a sample, the samples are actually composed of different cell types. So when you take a blood sample and you're measuring methylation in your blood sample, you're actually measuring many different kinds of white blood cell types are in that sample. So like, let's say you have orange cells, blue cells, and green cells. Well, if you have a person in your study and you take that blood sample from them, if you have methylation, you know, if at some place, at some CPG site you're interested in, if the green cell has 0% methylation and the blue cell has 100% methylation and the orange has 50, and you have those in equal mixtures, this person's mean is going to be 50% methylated, but their cell types actually look really different. And so we have to remember that we're getting a mean of what's going on within all these different cell types in the sample, right? And that's really important because when we find an effect, right, when we see that someone's CPG at some particular CPG is more methylated in some people than others, and that could be due to a variety of things, right? So let's say we're interested in some exposure of interest, and we're interested and we see a relationship between the exposure and methylation at this CPG site. Well, that could be because the exposure really did have an effect on methylation in all of the cell types, right? Had a direct effect. Maybe it increased the methylation. Increase the methylation in this cell type. Well, it can't. It's already at 100%. in this cell type it went up, and in that cell type it went up. And that person's mean methylation went up too. Okay, that could happen. Or there could be a vulnerable cell type. So this methylation actually stayed the same in this cell type, but the green methylation went all the way to the top, right? So it did have an effect on methylation, but only in one cell type. Or maybe this exposure changed the composition of cells in our sample. So maybe the exposure changed the white blood cell type distribution in our blood sample. And the blue is more overrepresented now, and it got rid of most of the green, right? These are immune cells we're talking about, white blood cells. If an exposure changes how your immune cells are distributed in the blood, you could also see a change in methylation patterns, right? So this is something we have to be really careful about when we're doing analysis. Epigenetic profiles also change with age and with environmental exposures, right? So this is showing a chromosome where they were staining things in yellow if they had similar methylation, and then either red or green if they had different methylation. And these are pairs of twins. So this is twins at 3 years old, and this is a pair of twins at 50 years old. And you can see that the 3-year-old twins, their methylation patterns looked really similar. And at 50, these twins' methylation patterns looked pretty different in a lot of places, right? So they have the exact same DNA, but their epigenetics changes over time, right? Those are divergent, probably due to different environments, but also due to the effect of like drift with age. Your epigenome like can't maintain itself as well with age. So your profile changes with age in a characteristic way. It also changes in like a a way in which it just is sort of falling apart. And then the lifestyle and environmental exposures are also going to increase that divergence with age. Cell proportions also shift throughout life. Okay. So those white blood cell types I talked about, they also change with age. So when you look at things like this is the proportion of granulocytes, one of those white blood cell types in the blood samples, that characteristically rises with age. So that's going to drive some of the observed age-related epigenetic differences that we see. So if we don't really take that into consideration in our analysis, it's going to confound our analysis essentially. And just another quick thing about timing is that exposures can be captured throughout the life course and even before. So for example, we don't transmit directly epigenetics for the most part from one generation to the next. So when I have a child, I transmit my DNA sequence directly from myself to my child, right? But our epigenetics doesn't really do that. There's waves of demethylation where a lot of those methylation patterns are washed out. However, when you have a situation where you have an exposure, remember that for example, a woman who's pregnant, her fetus is developing at the time. So if you have an exposure to a pregnant woman, her fetus will have that exposure as well. And the fetus is actually making germ cells at that time. So the eggs that will make the next generation, if this is a female, for example, are already starting to form. So it can affect multiple generations simultaneously, some of these epigenetic exposures. And there are also really critical developmental time periods. So throughout the life course, there are places where epigenetics is reprogrammed and kind of remade in your epigenome. So there's maternal imprinting established when you're a fetus. Then you have changes around adolescence that are important, that are epigenetically different. and during aging. And so there's these critical time periods where your epigenome is forming, where exposures could be particularly important. So there's these sensitive time periods where you might be especially sensitive to exposures and its impact on your epigenome. Okay, just going to give you a brief primer. This is one more section about why we want to study epigenetics in humans, for example. Okay, why? Well, it's critical for growth and development. It can help us provide biological inferences and understand the biology behind diseases. We can study the mechanisms of genetics on health, the mechanisms of physical and social effects on health. We can look at potentially interactions with genetics or environment. Epigenetics can be a biomarker of exposures and of aging, and then we can also help predict disease. So in a precision health context specifically, we can characterize these mechanisms by which our genetics and our environment lead to disease, right? We can understand more about molecular physiology and its relationship to genetic, physical, and social environment, and potentially develop new treatments and interventions. This has already been important for things like cancer. There are epigenetic treatments for cancer. We can enhance precision medicine and public health, so earlier detection of subclinical disease processes, so identify people at risk and identify processes earlier before they actually are causing disease. Better predict the course of disease and tailor and target interventions and treatments to the individual. Another thing is that understanding and characterizing how social and environmental influences affect the epigenome can help us advocate for policy change and give us a tangible thing to point to and say, hey, look, this is really affecting our biology. There's a lot of different epigenetic methods. We're going to talk about a lot of these, from hypothesis-based to discovery-focused. Summary measures, global measures, scores, and site-specific pieces. We're going to talk about all of these. Epigenome-wide association studies, where we look at CPG sites, for example, across the entire epigenome and look at their relationships to disease, have become incredibly important and form a foundation for a lot of the work we do in this space. So we're going to talk about EWAS in general, sort of the epigenome-wide association study. We're also going to talk about finding differentially methylated regions, where we kind of look for regions of the genome that are showing different patterns of methylation. The EWAS catalog tells you about research being done in this area. So there's 1,127 epigenome-wide association studies published in this curated EWAS catalog. About 800,000 associations between CPG sites and 837 traits of interest, about 4,000 cohorts and 230 tissue types. What are the things that are most studied? Smoking, that's huge, affects your epigenome like crazy. Aging, same. Body mass index and type 2 diabetes, also very big associations with the epigenetics. Then you get things like Alzheimer's disease, which is really interesting because it's in the brain. So it's hard to study in the blood. So a lot of these are like postmortem brain. Some of them are blood-based. But then two more measures of obesity, waist circumference, and gestational diabetes. Okay, so in brief, the reasons why here, it can help provide biological inferences. So if we find a bunch of epigenetic sites associated with a disease of interest, we can look at what gene sets they're in and see if they help identify specific biological pathways. So we can use the gene ontology and the Kyoto Encyclopedia pathways to see if there are underlying biological similarities to these genes and if they're part of some underlying biological process that can help us understand disease better. We can use epigenetics as a potential mechanism for these genetic effects on health. So if you've got a genotype of interest, and it influences a trait, like if you found something through GWAS, you can see if that genotype is actually influencing nearby DNA methylation, which can cause changes in gene expression, which are leading to the trait, right? So we get at that underlying mechanism of that genotype. So for an example, um, uh, so there was a study of smoking on DNA methylation and lung function. So they had genotypes that were predictive of smoking. And those genotypes were associated with lung function. They wanted to know, well, is it through smoking behavior and that changes methylation and lung function, right? So you can do these mediation kind of models and look at these pathways. And this was a study that did find that some of these CPG sites were actually acting in this mediating pathway between genetics and lung function. You can also use methylation as a tool to study the mediating effects between social or psychosocial impacts on health. So here's a study this is kind of some of the work that we've been doing, looking at things like socioeconomic status, education, income, wealth. That affects your cardiovascular risk, hypertension, diabetes, and obesity. Well, does it do it through changes in DNA methylation? So here we had tested for a mediation, which is this pathway here, and found that 7 CPG sites actually did have show some mediation effects. So they seem to be mediating about 20% of the pathway between socioeconomic status and health here. You can look at interactions with genetics or the environment, right? So this was a paper that looked at asthma, and what they found was in people had higher levels of methylation. So that's the histogram. The higher levels of methylation, they had a higher risk of asthma, but it was much bigger for people with this genotype right here. It's the CC genotype of some specific SNP in interleukin 4 receptor than people with this genotype, right? So if you had 2 copies of the C allele, you had a much higher risk ratio, but only if you had methylation also at this CPG site, right? So an interaction. Epigenetics can be really great biomarkers of exposure. It can help you recreate the past, right? Sometimes they're more accurate than current measures. So if you take, if you measure someone's blood cotinine level, so that's a measure of nicotine in the blood, basically, or their recall of smoking behavior from surveys, you can actually do a better job of recreating their past smoking history by looking at their DNA methylation. It's the same thing for environmental exposure. Some of these heavy metals, air pollution, maternal exposure during pregnancy, there are CPG sites that track that stuff very, very well. We'll talk a lot about epigenetic biomarkers of age and aging, but essentially you can use epigenetic clock methods to summarize whether you think people are aging faster than you would expect chronologically or slower. And a lot of those clocks are associated with health outcomes and with social and environmental exposures, including things like mortality, cardiovascular disease, and cancer. Epigenetics are also predictors of health risk factors. So this was a study, and we're going to talk about this too when we talk about polyepigenetic scores, where they looked at variation explained in BMI and height by a genetic risk score, so a summary measure of genetics, and a summary measure of methylation or a methylation risk score. Okay, and both of those in BMI explained variability sort of additively. So in BMI, you've got in red the methylation risk score, and in green the genetic risk score, and in blue both of them together. And you can see they're kind of explaining different pieces, right? Because when you add them together, they explain more. But with height, the methylation risk score didn't explain almost anything compared to the genetics. So different for different traits. And the last one is that epigenetics can also be predictors of disease. So this is, we're going to talk about machine learning. And this is an example where they constructed methylation-based subtypes of a kind of cancer based on methylation signatures. And they found that those subtypes predicted things like prognosis, recurrence, and immunotherapeutic implications, right? And so this is really big now, and why we're going to talk about making surrogates um, is that you can use it for prediction, right? And in a lot of cases, it does better than genetics ever did, for different reasons, which we can also talk about.